In a study published yesterday, August 5, in Science, researchers identify human and mouse long non-coding RNAs, whose loss mimics a metabolic disorder phenylketonuria, PKU, in human cells in mice. Injecting the human long non-coding RNA into a mouse model of the disease improved the animal's symptoms, potentially pointing to new opportunities to develop treatments for human patients. Scientists and clinicians have historically categorized PKU as a problem with the gene that encodes the enzyme phenylalanine hydroxylase, PA, which converts the amino acid phenylalanine into tyrosine. Without this enzyme, phenylalanine consumed as part of a normal diet in foods such as milk, eggs, cheese, eggs, and meat builds up, causing seizures, intellectual disabilities, and psychiatric disorders. But DNA sequencing has demonstrated that sometimes people with PKU don't have a mutation in the gene for PA, calling into question PKU status as a single gene disorder. The new study suggests the depletion of certain non-coding RNAs could also cause the condition, and that boosting the levels of those RNAs could relieve symptoms. This just shows that even after such a long time of studying what we thought was simple disease, we're still learning more and more about other factors than just the mutations in this specific gene, says John Mitchell, a pediatric endocrinologist at McGill University Health Centre in Canada who We haven't been able to really describe the genotype-phenotype relationship for this particular disease, he adds. Having new areas to pursue for treatment is going to be important. See The RNA Age, a primer, in the new paper. University of Texas MD Anderson Cancer Center's Lu-Ching Yang and colleagues write that nearly all mutations in the human genome happen in non-coding DNA. Based on this idea and on work showing that PKU sometimes occurs in the absence of mutations to the gene for PA, they sought to identify potentially relevant long non-coding RNAs by searching for ones with differing expression levels in mouse livers, the main site of phenylalanine metabolism, at 2p. Yang and his collaborators identified two versions of a non-coding RNA that were upregulated in adult mouse livers, suggesting they may play a role in phenylalanine metabolism. The researchers then introduced a mutation to disrupt the first splice site in the non-coding RNA, which depleted both forms. The mice with reduced non-coding RNA were slow-growing, had higher levels of phenylalanine in their serum, and displayed diminished PA activity, similar to people with PKU. Furthermore, as these mice aged, they experienced seizures and early death. The team used an antibody to isolate PA and determined that the non-coding RNA they found is associated with the enzyme, so they named it PAIR, PA-activating long non-coding RNA. In an email to the scientist, Nenad Blau, a consultant in biochemical genetics at University Children's Hospital Zurich in Switzerland and co-author on the paper, notes that HULC deficiencies have yet to be detected in patients with abnormally high phenylalanine levels. Still, he says the work highlights that PA genotyping is essential for the diagnosis and management of such disorders. It will be fascinating to see whether or not we can find changes in this long non-coding RNA or others that actually do explain some of the phenotypic variability seen in PKU in people, he says. The next logical question is, if there is variability in the long non-coding RNAs, is there therapeutic potential? See, new treatments for phenylketonuria aim to loosen reins on strict diet. In an effort to address that sort of question, Yang, Blau and their colleagues delivered a liver-targeted WHO control mice that received an injection of a scrambled RNA had high serum levels of phenylalanine, but treated mice had comparatively low levels of phenylalanine and high levels of tyrosine. Then the researchers gave HULC mimic alongside an established drug, a synthetic version of a protein known to support PA function, that works in some patients with mild cases of PA deficiency, but has been shown before to be ineffective in this mouse model and in patients with full-blown PKU. The combined treatment had an additive effect in the mice, lowering serum phenylalanine levels even more than HULC mimic alone. According to Blau, the results indicate that HULC mimics may be a viable treatment option for some patients with PKU. However, before applying a related strategy therapeutically in people, he notes that research would need to address whether it's possible to manipulate one enzyme without having negative effects that would harm the patient. And while Young, Blau, and their colleagues have identified these RNA candidates that appear to function in similar ways, he adds that another challenge with long non-coding RNAs seems to be how do you jump the species gap, because your model systems may or may not actually be accurate mimics of what. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.